Hi everyone, my name is Anika Shah, and I'm going to be talking about CRISPR screens and their applications. So what is CRISPR? The main takeaway from CRISPR is that CRISPR allows us to cut genes. So every organism is made out of cells, and within these cells, there's DNA, those nucleotides that essentially code for who we are and what we look like. And it's those nucleotides that have those specific genes. One might code for skin color or specific proteins that our body produces. And CRISPR allows us to take a gene of choice out. We're able to remove it from the DNA. And that's what makes it so special, because it's essentially a form of gene editing. So how does this work? So you have these single guide that correlate to the target genes. So it guides this Cas9 protein to the target gene. So if we want gene A, it'll guide this Cas9 protein to gene A. And these Cas9 proteins are essential to CRISPR because they are the ones that cut the genes. They are the ones that make the edit. So these Cas9 proteins are basically now able to remove the gene from the DNA makeup of a cell. So how does this work? What do I mean by single guide RNAs complement the target DNA? So what researchers do is they take the DNA of the target DNA that they want. Say they want this gene right here that I have. It says template DNA. Say that's the gene that we want. Then what researchers will do is they will translate it into RNA. RNA essentially has matching nucleotides. So the DNA is made up of a bunch of nucleotides: adenine, cytosine, thymine, and those are represented by the letters A, C, T, and G, which is the blue one. And what the RNA basically does is it has matching letters. So for every adenine, there's a U in RNA. For every C, there's a G. For every T, there's an A. So the RNA essentially matches the DNA. The DNA made the RNA, was the template for the RNA. So now the RNA matches the DNA. It just has a slight difference in nucleotides because that's just how it works. So now you have this matching RNA, and it will find that DNA that it matches with. And that's how it guides the Cas9 proteins to the target genes. So what researchers can do with this is they can take a set of genes, say you have, A, B, C, D, E, and F, and after that CRISPR-Cas9 editing, they're able to get different combinations. So now you have B, C, D, E, F without A, because they removed gene A. Now they can remove gene B. Now they have A, C, D, E, F. And then they can do the same thing, but remove C, and then et cetera, et cetera. And you basically get a bunch of different combinations, and each change that you make is altering the cell's genetic makeup. And sometimes those edits will have very minimal impact. Other times, those edits will have very strong and very heavy impacts on the cell and the cell's behavior. So how are these single guide RNAs created? So obviously for every researcher, every experiment, every lab, it's going to be different, but they all follow this sort of universal three-step process. So they have their DNA, as I mentioned before, that converts into RNA. It translates into that matching RNA that codes for the target DNA. So now you have these RNA that match your target DNA. Then these RNA are converted into plasmids, which are circular, and the reason is because you want them to be in this lentivirus plasmid form. That is because these single guide RNAs need to go through the cell membrane, they need to go to the nucleus, so you need a structure that will be able to go through all these different almost barriers, such as the phospholipid bilayer. So these lentivirus plasmids are able to do that. They're able to efficiently take the single guide RNA into the nucleus so that I can start cutting away at the DNA. So once you have that pooled lentivirus library, they're ready to be injected into whether that be cell or an organism. So that's sort of how these single guide RNAs are created. So what can you do with this? CRISPR has various uses, but the main one that I sort of studied was how CRISPR screens can determine target genes for drugs that fight diseases. I'm going to be talking about a specific application in cancer, since that's obviously very prevalent and oftentimes deadly disease. So what researchers are able to do is they're able to pool cancer cells together from, say, a patient's tumor. So now you have essentially like a petri dish of cancer cells. and they inject those single guide RNAs, but keep in mind these single guide RNAs all code for different genes. Oftentimes, sometimes, or most of the times, there will be like 100 single guide RNAs that code for gene A, and then 100 that code for gene B, etc., just to ensure efficiency and accuracy. But what these single guide RNAs will do is they'll go into this cancer cell culture and they'll start cutting away. They'll start changing. So see, now you have this plate from a plate of all of the same cells to a plate of different colored cells, because now each cell has a different genome because genes have been cut. The genome has been altered. The cells are officially different because they're going to behave differently, because your DNA, anyone's DNA, essentially codes for how you look, what you are. So those cells are also going to experience differences. And so now what researchers will do is they'll analyze how these cells behave. So how does the cell behave when it doesn't have gene A? Does it die out? Does it live? Which single guide RNA edit correlates to cells living or dying? And this is because, say you remove gene A, and all the cells that don't have gene A, all these cancer cells without gene A, start dying out. That means that researchers can try to use drugs to target that gene, because we know that the removal of that gene leads to the dying of the cancer cells, which is the ultimate goal, like killing the cancer cells. So that's why CRISPR is so cool, because researchers are essentially able to see how cells behave with different genetic makeups. And another application that's oftentimes more accurate, however, has its ethical qualms, is direct in vivo screens, which basically allow researchers to see how actual organisms behave when that S single guide RNA goes into the organism. Because for now, they've been doing it on these plates. But now what happens if you put it in an actual living thing? How does the living thing react? And how does the cells in a living thing react? This is obviously more accurate because these edits are going to be affecting an actual living person, not just on a petri dish. So now they do that same thing. They study the amount of single guide RNAs in that organism. Did the single guide RNA that cut gene A, is that amount going up? Because that means that the cells that have been cut, like gene A has been cut from those cells, are living. Or if the amount of single guide RNA goes down, that means the cells that the gene A was cut from are starting to die out. So they sort of measure the amount of single guide RNAs in the organism. And this allows the researchers to see how these cancer tumors react to different cell edits. And so one part of my project that I used was CRISPR. It was created and designed and coded by a couple of researchers from UCSF. And how it worked was I essentially downloaded Julia, which is a programming language. And using Julia, I was able to download CRISPR. So CRISPR was the program that I was running. And after exiting Julia, and then I ran the CRISPR program. So I have couple of images here. I downloaded Julia and then I ran CRISPR. And then what I essentially did with CRISPR was I edited the amount of single guide RNAs in a CRISPR screen, or the amount of cells. So basically CRISPR allows you to do a digital CRISPR screen, almost a digital model, an online model of how your CRISPR screen might go, with what numbers of single guide RNAs are you using, what number of cells are you using, and other factors, numerical factors that you may consider when calculating how many of what to use. So what researchers do with this is, because CRISPR screens are so expensive and so time consuming, they want to sort of optimize their results. So they use CRISPR to do that. So for example, on a dropout screen, researchers are looking for genes that are essential for cell growth or proliferation. So if a cell starts to die out when a certain gene is removed, that means that that gene was essential for that cell. So as you can see here, they're looking for that number in pink, decreasing. Which edit is making the amount of single guide RNAs go down? Because as I mentioned before, if the amount of a specific single guide RNA is going down, that means the cells with that single guide RNA is starting to die out. That means that this gene that they removed is essential to the cell's growth, and it's causing it to die out. Another thing that researchers might do and often a lot of them do, is put cells under different pressures. So what if you put a cell under this drug and take out this gene? Then what happens? What if you put it under the influence of another drug? So they're almost pairing two treatments together. What happens if we knock out this gene and put a patient under this medication? Then what happens? And that allows them to test all these different combinations. So yeah, that was my presentation on CRISPR screens and their applications. I hope this interests you because it is a very interesting and up and coming new field, and I'm very excited to see how it goes. Thank you.